Hi everyone. In this unit, we're going to be taking a closer look at excitability inside of neurons. So in order to understand this unit, you're going to have a pretty good grasp of the membrane potential concepts that we talked about in the previous unit. I'm going to start off by talking about neurons as a tissue, and I'm going to do that pretty generally because we'll be expanding on that in our later units dedicated to the central and peripheral nervous systems. The major focus for this unit is going to be to talk about membrane potential inside of neurons, and specifically about graded or synaptic potentials and action potentials. These are two types of changes in membrane potential that are required for the nervous system to function properly. So we're going to talk about how neurons use those changes in membrane potential to propagate signals between two neurons or between a neuron and some other tissue type inside the body. Now, as you probably know from previous courses, neurons are the basic unit of the nervous system, and they're pretty unique in terms of their function. They make up one of the body's major control systems. Their job is to propagate signals. But they also have some pretty interesting histological characteristics as well. If you remember back in the membrane transport unit, we were talking about the relationship between active and passive transport. And I mentioned that the sodium-potassium pump was critical for maintaining concentration gradients. That's because the nervous system depends on concentration gradients in order to function. So neurons have to have a very high capacity to generate ATP in order to keep the ions inside and outside the cell within homeostatic ranges. Neurons also tend to live longer than other cell types inside the body. That's especially true if we compare neurons to something like epithelia, which are constantly getting replaced. And as a terminally differentiated cell, a neuron doesn't go through mitosis. So this gives rise to a common misconception that neurons can't be replaced. You've probably heard someone, probably your grandparents, telling you that you're killing your brain cells by doing something that they don't like. It turns out that we can replace neurons. It's just kind of complicated because in order to do that, we have to go through that stem cell differentiation process. And so the problem really is when we go through that process, we can't get the neurons to differentiate in the right spot or to make the same connections with other cell types. So this is the reason why we haven't been able to make as much progress with respect to neurodegenerative diseases and traumatic brain injuries as we would have liked to up to this point. Now, you can see from the picture over at the right that neurons have a drastically different morphology than most other cell types inside the body. They tend to be very processy. So I want to really quickly introduce the different regions of the cell because I've chosen to organize this unit by focusing on what happens in different regions of the neuron. So the four major areas of a neuron are the dendrites, the cell body, the axon, and the axon terminal. And we can think of the dendrites primarily as a place of signal input. They contain really high densities of receptors because they're constantly receiving signals from external and internal environments. So all the signaling comes into the neurons for the most part at the dendrites. And so as you might expect, signaling at the dendrites is incredibly complex, right? If you take a look at the picture that's on the right-hand side of this slide, which is actually of the hippocampus, you can see the dendrites are highly branched and highly overlapping. And it's pretty much impossible in this picture to map a single dendrite back to the neuron that it originates from. So most of the neurons that I'll be showing you in this slide set, and actually most of the neurons that we talk about in this entire course, are multipolar neurons. And that means that there are many dendritic processes that are coming off of the cell body or soma. The only thing that I really want you to appreciate from the picture that's here on this slide is that that's an oversimplification. There are a lot of neurons inside your body that look pretty different than the cartoonized ones that we'll be seeing in this slide set. Now, some neurons have hundreds of dendrites, others have one or two, and some of them have no dendrites at all. So dendrites are projections of the cell that come from the cell body of the neuron. The cell body is also known as the soma. Now, for our purposes, the soma is not too interesting because this is actually where we find most of the things that we would typically associate with cells, like many of the cell's organelles, the nucleus, lots of mitochondria, so on and so forth. The neurons that we're looking at on this slide, you can see have a granular type of appearance. And the arrows are pointing to structures called missile bodies, which contain high densities of ribosomes. And that accounts for the large amount of protein synthesis that neurons have to do. Remember, though, that the soma is not the only place where protein synthesis occurs inside of the neuron. It can happen everywhere, even outside of the soma. Now, typically, the longest process that comes off of the cell body is known as the axon. So you can see from this picture that the soma tapers to form an initial segment of the axon that's also known as the axon hillock. That region is going to be pretty important to us a little bit later in the unit. And eventually, the axon hillock becomes the axon itself. Now, axons can be really short, like for example, in your brain or your spinal cord, but they can also be really long. So for example, a single axon might travel from the tip of your toe all the way into your spine. So again, just to try to represent this complexity, I'll point out that in some cases, neurons don't have an axon at all. And we'll see one example of that a little bit later on in our visual system. Now, axons eventually form axon terminals, which are important for forming synapses or connections with other cell types. Those can be other neurons or non-neuronal cell types inside the body. So if a neuron's going to synapse with another neuron, then most commonly it's going to synapse with the dendrites of that neuron. That's known as an axodendritic synapse. There's also something known as an axo-axonic synapse, which is where a dendrite synapses with the axon of another neuron. But we're not going to talk about that until a little bit later in the course when we get to our sensory unit.